Hello guys, welcome back to our YouTube channel. In this video we are going to discuss the regulation. So let's start. Accidental activation of HR through NLR proteins could cause vast destructions of the plant tissue. Thus, the NLRs are kept in an active form through tight negative regulation at both transcriptional and post transcriptional levels. Under normal conditions, the messenger RNA of NLRs are transcribed at very low levels, which results in low levels of protein in the cell. The NLRs also require considerable number of chaperone proteins for their folding. Misfolded proteins are immediately ubiquitinated and degraded by the proteasome. It has been observed that in many cases, if the chaperone proteins involved in the NLR biosynthesis are knocked out. So HR is abolished and NLR levels are significantly reduced. Intramolecular interactions are also essential for the regulation of HR. The NLR proteins are not linear. The NBARC domain is sandwiched in between the LRR and TIR by CC domains. Under normal conditions, there is a lot more ATP present in the cytoplasm than ADP. And this arrangement of NLR protein prevents the spontaneous exchange of ADP for ATP and thus activation of HR. Only when a virulence factor is sensed, the ADP is exchanged for ATP. Mutations in certain components of plant defense machinery result in HR being activated within the presence of pathogen effector proteins. Some of these mutations are observed in NLR genes and cause these NLR proteins to become autoactive due to disrupted intramolecular regularity mechanisms. Other mutations causing spontaneous HR are present in proteins involved in ROS production during pathogen invasion. HR is also a temperature sensitive process, and it has been observed that in many cases plant pathogen interactions do not include HR at temperature above 30 degrees Celsius, which subsequently leads to increased susceptibility to the pathogen. The mechanism behind the influence of temperature on plant resistance to pathogens are not understood in detail. However, research suggests that the NLR protein levels might be important in this regulation. It is also proposed that at higher temperatures, the NLR proteins are less likely to form oligomeric complexes, thus inhibiting their ability to induce HR. So this is all about today's video. Thanks for giving us your precious time.